study guide review. Okay. So step by step, I will not recap the entire lecture, for each topic, but mechanism for bacterial growth, only thing that you have to know is that it's what? Binary fission, right? The steps that are necessary. Cell has to multiply the DNA, replicate the DNA, and probably synthesize some additional proteins. That's pretty much it. Generation time. Yes, I'm going to ask you both ways. I mean, I can't tell you what exactly will I ask, but I'm going to ask you a question. Look, you have such and such generation time, so many cells in the beginning. How many cells are you going to have after 3 hours? Okay, you had one cell in the beginning, I don't know, 32 cells after 2 hours. What's the generation time? Okay, so you have to remember that with each generation time, what happens to the number of cells? It doubles. That's it. Not triples, it's not quadruples, it doubles always. So you have to remember, like generation time 20 minutes, number of cells doubles in 20 minutes. Does that make sense? So please understand that, okay? Phases of the typical microbial growth curve. Lag, logarithmic or exponential, stationary, death. 4 phases. Know what happens to the cells at each phase. Obviously, they all grow at each phase, but at some phases they grow faster, then they die, and at some phases it's the opposite. Okay, understand this. So if you have a question, cells die faster than they multiply, refer to death phase, obviously, right? Death and division rates are equal, it's stationary, and so on and so forth. Understand the availability of nutrients at logarithmic. More nutrients are available, in stationary, it's kind of wastes start to accumulate. Yes? No, wastes accumulate all the time. All the time, yeah. Because if you will take the liquid culture in the tube, we'll start sampling the density of microbial population. You will see definitely the logarithmic phase. And then since you don't take anything out, microbes will produce more and more waste and use more and more nutrients. So the balance will shift towards waste accumulation. Does that make sense?
Is there, like, between log lag, death, and so on, does one produce more waste than the other?
Oh, it's a great question.
Dying off, would that contribute to the waste?
I cannot tell you. I can tell you dead cells, they may contribute to the death of other cells, but essentially when cells replicate actively and grow actively, they produce more. But since they have abundant nutrients, they sort of don't notice it enough. When concentration of whatever toxic chemical you have, like ethanol, reaches a certain level that is incompatible with the growth, cells stop growing. And I would say that for each population, that would be different. Basically, so the phases of the growth curve will be the same for every microbe, but the length of each phase will be individual for each microbe. Does that make sense? Does that make sense? They're all going to go through the same phases, but the shape of the curve will be slightly different. Some microbes will grow for a longer time, but slower. Some microbes will grow faster, but for the shorter period of time. Okay. So yeah, understand the difference between the lab and the natural environment. We discussed that in nature, cells exist in a stationary phase, right? In the lab, it's mostly exponential or logarithmic. Understand the idea of persister cells that slow down the metabolism and capable to survive through the lack of nutrients. And your understanding of chemostat. What I expect you to know is that that's the special kind of growth vessel that allows for constant removal of wastes and supply of nutrients. So cells can be maintained in the logarithmic phase all the time. Does that make sense? That's all you need to know about chemostat. That it allows for the continuous growth. I'm going to put it here. Okay. Sorry, this pen, I don't know what happened to it. It's totally misaligned. So it's really weird. I have to write like an inch to the right to the actual writing. So that writing can be awkward. Okay. So continuous growth. Measuring bacterial growth, we discussed it. You have to understand the basic principles, whether we're counting viable cells or total cells, whether we're counting cells at all, like in case of turbidimetry, we don't. We measure optical density. Okay, so be able to tell apart. Like if I give you 4 choices and ask which method actually counts number of live viable bacterial cells, you have to tell me that. Does that make sense? I won't ask you, like, describe me the intricate technical details of the Coulter counter. I couldn't care less. Does that make sense? Good? Any questions so far? Sources of major nutrients, location and function in bacterial cell, define essential. Okay. Essential nutrients, something that cell cannot grow without. That's as simple as that. Bless you. Sources of major nutrients. Understand that oxygen mostly is acquired from the air, that level. Carbon, dead or live organic matter. In some cases, carbonates. I'm not going to trip you over on that, believe me. Seriously. If I ask you where do we take hydrogen, pretty much all you have to know is that hydrogen is H, and you have a selection of water, sulfur, carbon, and oxygen. You have to understand that H, which is hydrogen, is only in water. Okay? Believe me, it's what I really want you to understand is where microbes use those elements. So if I ask you, in which molecule we're going to find sulfur? It's going to be protein. In which molecule we're going to find nitrogen? It's going to be protein. It's going to be nucleic acids, right? It's going to be some modified glycans, nitrogenated glycans. So you have to understand that. Any questions about this? Transport. We went through this so many times. Basic principles. Requires energy, does not require energy. Goes directly through the membrane or requires a transmembrane protein like a carrier or a channel or a pump. Okay? Against gradient, along the gradient. Does that make sense? Osmosis. Understand where water goes from low solute to high solute. Okay? Understand the concept of tonicity. Okay? Isotonic versus hypertonic versus hypotonic. And remember, these are relative terms. Okay? If the environment that surrounds the cell has lower salt concentration than cell itself, it's hypotonic. Absolute concentration doesn't matter. Does that make sense? Transport of large molecules, so for endocytosis, understand the difference between phagocytosis, pinocytosis, and receptor-mediated endocytosis. Pinocytosis is completely unspecific. Phagocytosis is somewhat specific. Receptor-mediated endocytosis is highly specific. Remember, in receptor-mediated endocytosis, you have a protein on the surface of whatever's being taken into the cell that interacts with the receptor. Does that make sense? Got it? Understand what exocytosis is. Exo. Transport of things outside of the cell in the vesicle. Period. That's all you have to know. Did I make myself clear? Excellent. I'm trying to figure out how should I position this damn thing. I think I found out. Okay. Bacterial communities. Symbiosis versus non-symbiosis. Symbiotic relationships. What is that? What's the difference between symbiotic and non-symbiotic? Symbiotic is the must, right? What types of symbiotic relationships, you know? Mutualistic, commensal, parasitic, right? The subtype of commensal is satellitism. That makes sense? Again, you will not have to distinguish between, say, satellitism and commensalism because one is the type of another. Okay, but I will ask you 1-2 questions about this. Which two types of non-symbiotic relationships we discussed? Synergism and antagonism, right? In this case, obviously in antagonism, microbes don't need each other, but compete and kill each other if they are adjacent. In synergism, they can grow without each other, but benefit from each other's presence, right? Understand like the basic definition, okay? You may see the actual definition or you may see the example of relationships, okay? Like microbe A supplies an essential nutrient for microbe B. Microbe A can grow without microbe B. How do we call this? That's going to be commensalism or it's going to be satellitism. You won't have to pick from these two. Does that make sense? Okay. Biofilm. What you have to know about the biofilm formation and biofilm growth. So first of all, planktonic versus biofilm. Planktonic is like liquid culture that you saw. The primary colonizers produce extracellular polymeric substances that make everything sticky, protect the primary colonizers from desiccation and immune response, and then secondary colonizers stick in and the biofilm grows. That's the very basic idea, okay? So understand the role of extracellular polymeric substances and understand the role of quorum sensing molecules, okay? That they signal, they regulate the density of bacterial population in the biofilm. Oh yeah! Moreover, it's pretty much always different species, but they can communicate with each other using quorum sensing molecules. Yes? Microbe population density in the biofilm, okay? Does that make sense? So you can, and then when I ask you about the benefits, you have to, kind of appreciate protection from immune response from the host, protection from desiccation, protection from antibacterial agents, nutritional cooperation. One member of the biofilm can provide nutrients for another. Right? Good? Saprobes and parasites. One saprobe, it's dead flesh. Well, not flesh, organic matter. Parasites eat live organic matter. Alright? Differentiate ecto and endoparasites. Ecto live outside, endoparasites live inside. Understand the idea of obligate versus non-obligate parasite, and the idea of the intracellular versus non-intracellular. 5 major types of microbes. That's I want to go over with you, because that's probably the hardest part in microbial growth. So let's do this. Holy moly, it's absolutely out of alignment. Okay. It's very weird. If you would see where the pen is, it's just awful. Absolutely horrible. So that's the tube, or believe me, it's a tube. Obligate aerobic bacteria. Where would you see the growth? At the very top, okay? That's your obligate aerobe. Obligate anaerobe. At the very, very bottom. Very good, okay? Microaerophile. Certain area of the tube where the concentration of oxygen is just right. Okay? Air tolerant anaerobe. Everywhere. Yes. Anaerobe is air tolerant, so it can grow everywhere, it doesn't care. Okay? Facultative anaerobe. Very good. Mostly at the top. For facultative anaerobe, mostly at the top, okay? But it will also be down here. Why the facultative anaerobe is going to grow better at the top while still growing throughout the entire tube? Go ahead. Okay, but why more at the top? Oxygen, what does it provide? What does it enable? What kind of metabolism? Cellular respiration, which produces more ATP, so they can grow faster. Does that make sense? That's why facultative anaerobes. So essentially you have to understand that facultative anaerobes are essentially aerobic bacteria that can do fermentation. Does that make sense? Brilliant. So these 5, I will ask you about this. I promise. Seriously, know the difference. Again, you may get the generic definition, sorry, or you may get a description of the microbe. Blah, 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 blah can do this. Like Clostridium botulinum can be successfully destroyed by oxygen, grows in entirely, well, it's kind of boring, you know. But you get the gist, right? So I'm going to give you a description of a certain microbe, and you will answer what it is. Does that make sense? What are the capnophiles? What are the capnophiles? What do they need, capnophiles? CO2. By the way, here, when it asks compare and contrast metabolic differences, you have to understand that, say, anaerobes cannot do aerobic respiration, right? Does that make sense? Like, aerobes can do aerobic respiration. Functions of peroxidases, catalases, and superoxide dismutases. What's the difference? Why these enzymes are needed? What's the importance of these 3? In which microbes are you going to find them? Correct. The microbes that can grow in the presence of oxygen, whether they use it or not. Does that make sense? So these 3 enzymes, peroxidases, catalases, and superoxide dismutases, neutralize reactive oxygen species. Obligate anaerobes do not have those enzymes. Does that make sense? Okay, that's the answer to how presence or absence of these enzymes defines the oxygen tolerance. 3 types of microbes based on the pH requirements. That's boring. Sorry. Understand which ones are clinically relevant. Mostly neutrophiles. Temperature requirements. Can anyone name all 5? Okay, can we all, as a hive mind, start naming those 5 based on the temperature without looking in the notes? Huh? Okay, speak up. Thermophile. So they grow at the temperatures ballparkish 40 to 80. I'm not going to ask you numbers, don't worry, okay? Don't worry. But if I ask you about the microbe that successfully grows at the boiling water, don't call it a mesophile, please. It's not, okay? So thermophiles, just under the boiling water, extreme thermophiles, boiling water and above, okay? What is the mesophile? Body temperature, normal temperature. Does that make sense? Okay. What is this psychotroph? Not. Well, does it prefer the fridge or it can grow there? It can grow. Normally it grows at the room temperature, but it can slowly grow at about 4 degrees Celsius. That's psychotroph. And psychrophiles, they grow in the cold, right? Does that make sense? I want you to tell them apart more of a qualitatively rather than quantitatively. It may sound very unclear. What I mean is, if I'm going to give you a description without a number. Does that make sense? Something grows on ice. It is psychrophile. Something grows at the body temperature. It's the mesophile. Does that make sense? Clinically important microbes are, you're good, mesophiles. Extreme, boiling and above. But again, look. You're all worried that I'm going to give you something about boiling temperature and you will have to pick between thermophiles and extreme thermophiles. No. I won't do that. Huh? Absolutely. Because look, I can make a successful argument that it's not a good question. If I can make that, I shouldn't give you that question. Does that make sense? I want you to have a conceptual understanding rather than go into the minute, intricate details, whether this is the thermophile, extreme thermophile. Who gives a crap? Other environmental conditions that may influence growth. Barophilic microbes, pressure. Xerophilic microbes, dryness. Polyextremophiles. Sometimes there are microbes that are called blah 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 extremophiles. Those are the ones that can tolerate a whole bunch of rough conditions. Make sense? What I want you to kind of specifically focus, understand the difference. Osmophiles are the microbes that grow in osmophiles. Concentrated solutions. Concentrated solutions. Now, halophiles are the microbes that grow in concentrated salt solutions. So, halophiles are the type of osmophiles. Is that clear? Okay? We good? Going further to control of microbes. Protocols for growth. inanimate surfaces, disinfection, sterilization, sanitization. 3 protocols. Does that make sense? Understand the difference. Which one takes care of everything, which one doesn't, which one pretty much uses mechanical removal of microbes. Okay? I will be as explicit as possible in my questions on that. I promise I will read through that a million times. Okay, and you're more than welcome during, if anything is unclear, you know the rule of the house, you can come and ask. 2 techniques for treatment of the live tissue: asepsis and dedermation. That makes sense? Good. Let me see. Okay, 4 levels of biological safety. Again, this is the tough one for me rather than for you, because I have to describe very specifically what I want you to figure out. Bless you. Find out. If we have minute doubt, I will take it into account. I mean, if you will start arguing with me whether influenza is BSL-2 or BSL-3, yes, we can do that. If you will try to convince me that influenza is BSL-4, I won't buy it. If you will try to convince me if it's BSL-1, I won't buy it. Okay? And things like, if you try to convince me that we can work with Ebola in this room on the table, I won't buy it. That damn thing. Okay. Resistance of the microbes. So things that you can really find on the exam. I will definitely ask you, compare the resistance. It can be pick the most resistant one, pick the least resistant one, or pick the sequence. So if I tell you that in my sequence, enveloped virus are most resistant, endospores are least resistant, that is bullshit, okay? Again, I'm not going to ask you what is more resistant, non-enveloped virus or asexual spore of the fungus. It's incorrect. I mean, it's not just. That makes sense? But if I ask you to compare endospore with anything else except prions, there's no question. Okay? I will be extremely explicit, separated. Does that make sense? Am I clear? Factors that may influence efficiency. This is more for a say question. If I ask you what would you rather use to sterilize, I don't know, the surgical suit, you will have to not only propose the method, but explain why. Okay? So if you suggest that you're going to use autoclaving to sterilize the IV solution, you have to make a damn good argument, because definitely the solution is going to change after autoclaving. You have to make a really good argument to convince me. Okay? But if you will say, look, we can use filtration, but we won't get rid of viruses. That's a good point. So this is more to prepare yourself for say questions, to keep in mind what different things will do. Like if you propose to use bleach to clean up the entire hospital, unless you have a lot of hobbits that will work for food, okay, then it's not a good idea. You probably should use something else. That make sense? Cytostatic. I don't think it will cause any problem. Okay. Cell wall, cell membrane. So what can we do with cell wall? We can destroy it. Heat, oxidants. What can we do with cell membrane? We can destroy it. Surfactants. What can we do with proteins and nucleic acids? We can denature them. It's a whole bunch like phenols and ethanol, and they all can do that. Steam. Moist and dry heat. What I want you to remember, that table that I have on the slide. Moist and dry heat. There's I think Chastity is right there. Yeah, like the big one. Yeah, the big table with the temperature methods. Understand, for each method, know the level of treatment. If I ask you if pasteurization can sterilize, you tell me no. Does that make sense? I'm not going to stop at every method, but understand this stuff. Like if I ask you, can we truly sterilize by freezing something? No, we cannot. And so on and so forth. I don't want to go through that. Again, when I say applications and limitations, what I mean is, if you use heat, you have to treat something that is heat resistant. I mean, patient is not that heat resistant. For example, okay? Heat, like food is not. Many chemicals aren't. Do you understand? Good. Same goes for radiation. Right? So 3 types of penetrating. The ionizing radiation. The most penetrating are gamma rays, then you have X-rays, then you have cathode rays. Good enough. Non-ionizing UV light. We generally don't use X-ray and we don't use cathode because gamma is so easy to handle actually. So pretty much you're comparing gamma and UV. Again, applications, people. What you use them for, those methods. Mechanisms. So gamma rays, single and double strand breaks in DNA, UV light, modification of the thymine residues, pyrimidine residues. Okay. Does that make sense? What's the next? Filtration. I try to make it sound smart. Mechanism of filtration. Filter is the bunch of pores. Small things come through, large things don't. That's it. That's the entire thing about mechanism, period. Nothing fancy. Understand that the size of the pore will define the level of control. If you can make a filter that is small pores, very small pores, that will stop even viruses from going through, you're good. That will be sterilizing. It's really hard, but you can do that technically. Okay? Generally, it's disinfecting technique. We capture the bacteria. Does that make sense? Again, applications and limitations. Air, fluids. Especially large volumes. Especially air. You can hardly autoclave it. Halogen-based disinfectants. What is the mechanism? Nobody will tell. Yeah. For halogens. Chlorine, iodine. They oxidizing. They are oxidizing, remember that. Chlorine and iodine, yes. They are oxidizing. Can they sterilize? Unless you pump the entire room with chlorine gas, they can't. Phenol-based disinfectants. Mechanisms, inhibition of the protein denaturation and membrane disruption. Okay, and remember we have had this case of triclosan. be able to explain why triclosan was not effective. Bisbiguanidins, that's a fancy word for chlorhexidine. That's what you gotta know. Chlorhexidine. Effective disinfectant that can be used in the surgery, OBGYN, nursing homes, and so on and so forth. Okay. Alcohol-based disinfectants are pretty much, this study guide goes slide by slide. Alcohol-based, peroxide, and surfactants. For alcohol-based, mechanisms of action, denaturation and membrane disruption, level of control, disinfection. For peroxides, we talked about hydrogen peroxide, we talked about peracetic acid. They can be sterilizing in a higher concentrations. For surfactants, two types, cationic and anionic surfactants, and why cationic are more effective than anionic. Okay, because they bind to the cationic that are positively charged, effectively binds to negatively charged surface of the bacterial cell. Heavy metals, you won't have to list. Okay, you have to recognize that, like, copper is the heavy metal, or gold, or zinc. Understand the concept of oligodynamic action, that they work in the minute concentration. Understand the limitations. They're toxic, expensive. Let me say a few words about these guys, the alkylating agents. Alkylating agents, gaseous, ethylene oxide, propylene oxide, formaldehyde. Liquid, formalin, which is formaldehyde solution in water, glutaraldehyde, beta-propione lactone. Understand what they do. They chemically modify proteins and nucleic acids, changing their structure and function, rendering the cell dead. Can they sterilize? Yes. Does that make sense? So one of the approaches that may not be a bad idea to kind of get yourself more ready for that, put the word sterilization and write down the methods that can sterilize. Because that will put methods in the big groups. Does that make sense? Like, what can sterilize? Autoclaving, incineration, peroxides, alkylating agents, gamma rays. Did I forget anything? Like, I'm talking hardcore. Technically, again, chlorine gas, but we don't want to be like Germans in the World War I. Okay. That's it. So those 5 things. Then you do disinfection. Like soap, like regular soap, will not even be in a disinfection because it's so mild. So you would go with most of the temperature controls, UV light, surfactants, phenols, alcohols, halogens. What did I forget? Yeah, it's pretty much bisbiguanidins, like chlorhexidine, that's it. And then degramation, it's pretty much soap. Degramation, sanitization. That makes sense? Okay, any questions so far? Drugs. I asked you about history. That table in the very beginning, that's what you have to know. I don't care about data, like which year Fleming got his Nobel Prize. I don't give a shit. But you have, I think it is important to know that. It is important to know that Fleming discovered penicillin. Okay, I want you to know that. Waxman discovered streptomycin. Pretty 2 names that I'm going to focus: Fleming and Zellman Waxman. discoverers of natural antibiotics. I don't expect you to memorize the names of the Germans. The Germans, for God's sake, it's almost impossible to memorize those last names, right? I'm Russian, I can understand that. But know that, like, first synthetic antimicrobial, sulfonamide. Okay. Fleming discovered what? Penicillin. Does that make sense? That table will be able to answer questions about it. Concerns. Cidal versus static, route of administration, broad spectrum versus narrow spectrum. What else did I forget? Anyone remember? It will define cidal versus static. Well, pretty much you can think about the therapeutic index, how toxic it is. Yeah, side effects. So think about this, okay? Like if I ask you which drug would you rather use for this predefined infection, you should pick narrow range rather than broad range. Does that make sense? Cellular targets for antimicrobial drugs. Can you name them? Can you start, please? Membrane. Cell wall. Huh? The ribosome. DNA and RNA synthesis. Fifth one. Metabolic pathways. Okay. So 5 like big class targets. Does that make sense? Okay. Now, be able to provide drug examples for the target. Okay. Cell wall biosynthesis, we have 3 classes. It's the penicillins, inhibitors of transpeptidase. We have vancomycin, which inhibits transglycosylase, and we have bacitracin, which inhibits the transport of the components. Okay, so these 3 classes. I wouldn't focus that much on the broad range versus narrow range. It's not a pharmacology class. Okay, but understand the mechanisms. Penicillins prevent formation of linkages between the glycan chains. Vancomycin prevents the formation of actual glycan chains. Bacitracin prevents the transport. Is that clear? Okay. Protein synthesis in bacterial cells, actually, I think it's okay. So we have aminoglycosides and tetracyclines that bind to 30S subunit. So aminoglycosides, they change the shape of 30S subunit, right? Tetracyclines prevent the binding of tRNA. And then we have macrolides and cosamides, chloramphenicol, and what did I forget? Oxazolidinones. Okay, so this 4 drugs that bind to the 50S subunit. Okay, for them, know the mechanism such, for instance, like chloramphenicol prevents the formation of the peptide bond. Okay, oxazolidinone prevents actually formation of the entire ribosome. Lincosamides cause the early dissociation of tRNA. It's more of a, what they inhibit. Do they inhibit the protein synthesis? Do they inhibit the cell wall synthesis? Okay. I will get to the resistance thing. Don't worry about that. We will discuss what you have to know about resistance, okay? I think Julia gave me a good roast last class trying to take out what I'm going to ask about resistance. Okay, I'm going to remind you all. Okay. Membrane. What do you have to know about the drugs that inhibit protein synthesis? I'm going to ask you why they have such a good specific toxicity. Why? Why drugs that inhibit bacterial protein synthesis are highly specific against bacteria, not against humans? Ribosomes are different, yes. 70S versus 80S, so they have very different ribosomal structures. Does that make sense? Understand this. Drugs that disrupt cellular membrane. We have polymyxins and we have daptomycin. I'm not focusing so much on the mechanisms, but what I want you to understand, why these drugs are so toxic for humans. Yeah, because membrane is the same, right? Membrane in bacteria and membrane in humans are the same membranes, structurally. So they're very non-specific. DNA and RNA synthesis. Which drug class of drugs inhibits DNA synthesis? Fluoroquinolones. Cipro, right? For this, I want you to know the mechanism. It binds to DNA gyrase or DNA topoisomerase. It's the same. Those are synonyms. Okay? It's like Margaret and Peggy. The drug that inhibits RNA synthesis. Rifamycin or rifampin. Rifamycin is the class of drugs. Rifampin is a name of the drug. Okay? They bind to which enzyme? If they inhibit synthesis of RNA, they bind to RNA polymerase. Very good. Okay? Understand that. Right? Why they are specific. Why they have good selective toxicity. Why they toxic to bacteria, not to humans that much. You have, I know it's painful, but you have to remember the molecular biology of the cell. Bacteria are eukaryotic or prokaryotic? Prokaryotic. Humans are eukaryotic, right? So transcription and replication in humans happen in the nucleus. So these drugs have to be at least delivered to the nucleus. And this is on top of structural differences between the enzymes. Does that make sense? Okay. Metabolic pathways, okay? We discussed we technically discussed three, okay? One that we discussed in excruciating details, metabolic pathway. Synthesis of what acid? Folic acid synthesis. So sulfonamides and trimethoprim inhibit two different enzymes of the same metabolic pathway. Does that make sense? Are we good on that? Is that understood? We good? We also discussed linezolid, the inhibitor of mycolic acid synthesis. That's all you have to know. But you do have to know, if I say linezolid and I say mycolic acid, which bacteria does it inhibit? Mycobacteria that require mycolic acid. And we briefly touched on diaryl quinolones, the ones that inhibit ATP synthase in the mycobacteria. Major mechanisms that bacteria use to resist antibiotics. Now, don't look. Let's figure it out. That's logic. Okay. what bacteria can do to prevent drugs from action. Okay, wait. Okay, so do enzymatic bypass. Change the target of the drug. So pretty much kill the drug. Pump it out. Mimic. Okay, produce something that will, I don't know if I shared this analogy with you. I'll share it in there. So we have more. Stop it from getting in, right? Just don't let it in. There's actually another one. They can simply overproduce target. There's so much target, the drug doesn't work. I've got an analogy, I was very proud of it. It was awful, absolutely horrible analogy for mimicry. It's like dog humping your leg. The dog thinks it's another dog, but it's not. Okay, yes.
So when it produces,
Mm-hmm.
drug is trying to bind, but you're overproducing the target site.
Yeah. So it's just,
So does that reduce the sensitivity of the drug itself, or is it just making it?
say, drug inhibits the enzyme. Hypothetically, you have sulfonamide that inhibits the dehydropterate synthase, the first enzyme in folic acid synthesis. You have 100 molecules of the drug, and that will inhibit 100 molecules of the enzyme. If you produce 10,000 molecules of the enzyme, that's like 1%. Nobody cares. Does that make sense? That's the entire idea. Just brutally overcome. Does that make sense? So this is what you have to understand about resistance in terms of the mechanisms. What I want you to understand as well is resistant bacteria, okay? Common resistant bacteria. Can anyone name them without looking? VRE, vancomycin resistant enterococci, MRSA, CRE, carbapenem resistant enterobacteriaceae, the drug resistant mycobacteria. It can be multi-drug, total drug, extensive drug resistant mycobacteria. Extended spectrum beta lactamase present gram negatives. Okay? We're going to find them, except for mycobacteria. Hospitals. Pretty much except, so MRSA, both hospitals and community, everything else is hospital. Tuberculosis, it's a community thing, okay? You acquire it not in a hospital, everywhere else. Does that make sense? Now, when we talk about resistance, what else I want you to understand is that it's a genetic thing, okay? That when we say microbe is resistant because it pumps the drug out, what actually pumps the drug out? What do we call the molecule? The question contains an answer. The molecule that pumps the drug out is called a pump. Yes. This pump is what type of biological molecule? It's a protein. And protein is encoded by a gene, right? A DNA gene, right? So this gene can be spread through the population. That train of thought, I want you to have this train of thought. Okay? We have a gene for resistance. Gene or genes. Okay? Does that make sense? There you go. So you have these genes, they can be spread through the population, and resistance can be acquired by other microbes. What drives the resistance? What drives the evolution overall? Just how we call evolution, when something evolves? It evolves in response to, it adapts to selective pressure. Or change. Well, when you say change, what kind of change? Changing environment, right? When environment changes, organisms adapt. You have to understand that. Okay? If I ask you why resistance develops, I want to hear about evolution. I really want. I want to hear that microbes adapt to changing environment, and we change that environment. Okay, does resistance occur naturally? Yes, because microbes employ warfare to fight each other. Efficiency of antibacterial drugs. Kirby-Bauer test, the one that we did last week. Dilution method. As simple, so you dilute the drug and see which dilution is effective. E-strips. And understand the idea of therapeutic index. What is therapeutic index? Quantitatively. In terms of numbers, it's a strict number. Ratio? Toxic dose? Exactly. Excellent. It's a ratio of toxic dose versus effective dose. So which index is better, high or low? Higher. Period. That's it. Is that clear? We good?